Good morning to all. Welcome to the Diamond Group of Institutions Neet and K-Cet online classes. As usually, today's class it is plant breeding. So some of the questions from the plant breeding is: read the following statements and select the correct option. Options are: A. Allopolyploidy is a type of polyploidy which there is a numerical increase of same genome. So here, out of the four options, which are the options or which is a statement, it's correct, we have to identify. So option A, allopolyploidy. It's a type of polyploidy. Polyploidy means in a cell sometimes has many number of chromosomes or more number of the chromosomes are present. So allopolyploidy in a cell. So you can imagine about the cell. Most of the cell is contained as a two sets of the chromosomes. Otherwise, in the nucleus contained as a single nucleus is contained. That single nucleus is contained as a two sets of the chromosomes. So more than two sets of the chromosomes are present. We call as polyploidy. So those are the chromosomes are they belongs into the different genome. If we are doing as the hybridization mechanism, otherwise we are doing any genetic engineering mechanism, those are the chromosomes are we are going to transfer from the different genome or different organism into the cell. So option A, allopolyploidy is a type of polyploidy in which there is a numerical increase of same genome. So numerical increase of the not a same genome, it's a different genome only. So option A is incorrect statement. And option B, micropropagation is a difficult to expansive method for plant propagation. So micropropagation is application of tissue culture. It's a easiest method to the production of plants within a short period of the time. But option they given as its expansive method for the plant propagation. It's not expansive method, it's the easiest method to the production of new plants. And option C, protoplast technology has opened up a avenue for the development of hybrids of even asexually reproducing plants. So protoplast technique. Here you want to know the protoplast culture. In the tissue culture, one of the application method is somatic hybridization. In the somatic hybridization, we are going to using as a two different plant species cells. Cell wall we are going to remove only protoplasm. Only protoplasm of the two cells will be going to fuse together. Will be get into a single cell. And these are the single cell are the two nucleus. One of the nucleus will going to degenerate, another one will be going to present, and they can produce into the new cell. That is only called as a somatic hybrid. So option C given as protoplast technology has opened up avenue for the development of hybrids. Hybrids means two different characters are we are seeing in a single cell or single plant or single organism of even asexually reproducing plants. In the asexually reproducing plants, we are creating into the or we are going to perform into the somatic hybridization, otherwise protoplastic fusion to the production of new plants. So option C is correct. And option D, artificial seeds cannot be directly sown in the soil like as in natural seeds. So artificial seeds also we can easily directly sown in a soil to the production of new plantlets. So out of the four options, option C, protoplast fusion or protoplast technology or somatic hybridization mechanism, it is used into the production of new plants. So option C only is correct. Coming to the next question. In order to obtain virus-free plants through tissue culture, their best method is. So in order to obtain virus-free plants through tissue culture, the best method is, options are there means through the tissue culture we have to create or we have to produce virus-free plants. So options are meston culture, protoplast culture, embryo-based and the anaerobic culture. So here you want to know virus-free plants means. Virus-free plants. If the plant is infected virus, from the plant we have created into the new plantlets. So the new plantlet should be virus-free. At the time we are going to using as a explant, that explant material should be options are meston culture. So meston culture, you know the meristematic tissue. Meston culture means meristematic tissues are we are going to use into the explant. So mesematic tissues are they can show continuous production of new cells within a short period of the time. So by taking our mescum culture, otherwise mescamatic tissues as explant to the production of new plants as a virus-free plants is easy. Otherwise, we are going to use into the production of virus-free plants through the mescum culture only. Because why we are going to using as a mescum culture only? You know the mescamatic tissues are they shows the continuous divisions. So compare the division of virus, cell division will be going to fast. So that's why those are the tissues or the cells which are there located in the mescamatic region. They don't have any viruses. So that's why we can use as a mescamatic tissues only or mescamatic culture only to the production of virus-free plants. And option B, protoplast culture. Now only we are going to discuss as a protoplast culture and embryo wrist. So at the time of tissue culture, we are going to produce the immature embryo. Through the tissue culture, immature embryo we are going to produce means that is called as an embryo wrist. And anther culture. So instead of the mescamatic tissues, otherwise any plant materials, other plant materials, we are going to use into the production of new plantlets. As an explant, we can use as a anthers. So through the anthers, we can producing as a haploid plants. Haploid plants are we are going to produce in the anther. Through the anther culture, we cannot produce the virus-free plants. So out of the given 4 options, mescum culture only is correct. And option next question is gene responsible for dwarfing in wheat is. So which is a gene is responsible for the dwarfing in wheat? In the wheat plants, which gene is responsible for the dwarfing? Option A, D, G, O, G. So these type of the genes are we are seen in rice plants. So to the production of semi-dwarf high yield varieties of rice, these are the D, G, O gene was exported, introduced into the India from the Taiwan. So the gene name is D, G, O gene. And option B, Norrington. To the production of semi-dwarf variety of wheat by the presence of this Norrington gene. This is a gene name. Norrington gene will be going to use into the production of semi-dwarf variety of wheat. Wheat plants, semi-dwarf wheat varieties is developed by the presence of this Norrington gene only. And in the same way, like as the wheat, instead of the wheat they given as a rice, we will going to get the answer DGO gene. This is the gene name we are seeing in a rice. And option C, trigene. These are the crystallized genes we can see into the BT cotton. So those are the specialized genes collected from the Bacillus thuringiensis bacteria. And option D, not inducing genes means we can see into the rhizobium bacteria. Root nodules are they have to form by the presence of specific genes. Those genes are we call as nod factors or nod gene. So these are the genes nod genes are not used into the production of dwarfing of wheat. So out of the given 4 options, Norrington gene only will be going to use into the production of semi-dwarf wheat variety. The next question, polyethylene glycol method is used for option A, biodiesel production, option B, seedless production, energy production from seedless and formation of somatic hybrid. See now, polyethylene glycol is a chemical. It is also called as sodium nitrate. See now, this polyethylene glycol chemical they are going to use into the somatic hybridization. The mechanism of a process of somatic hybridization, we are going to use it as a polyethylene glycol. They are also called as PEG. So what's the use of this polyethylene glycol? So like as a hybridization, 2 different plant cells, protoplasm, we are going to combine together by the presence of PEG, the presence of PEG, and then form into a hybrid variety. That hybrid variety can act as a new variety of plants. So now polyethylene glycol method is used for the production of somatic hybrids. So those are options A, biodiesel production. For the biodiesel production, this polyethylene glycol is not used, and seedless fruit production. production of seeds without seeds, production of fruits without seeds occur by the phytohormones. Those are the phytohormones or genes and gibberellins will be going to use for the production of seedless fruits. So these are the seedless fruit production. Also the production of seedless fruits also this EEG will not be used, and energy production from the seedage. So energy production from the seedage, you know about microbes in human being, microbes in human welfare. So those are the microorganisms are used in a sewage treatment. What are the waste material they are to use, will be collected or they are separated. That waste material will be used for the production of biogas. That biogas is used as a energy for the lighting process. So here these are the sewage treatment also, otherwise production of energy from the sewage also. This chemical is not used. So C option also incorrect, and remind option D, formation of somatic hybrid. As I said, it is polyethylene glycol will be not used into the somatic hybridization mechanism. Somatic means vegetative cells. So these are the vegetable cells are used into the production of hybrids. Means two different characters are combined together and produce a new variety of plants. That is we call as a somatic hybridization. It's an application of tissue culture. So answer is next. Pusa snowball K1, an improved variety of cauliflower has been developed. So we know in the plant breeding, some of the disease-resistant varieties are being developed. So those are the disease-resistant varieties are Pusa snowball K1, one of the variety of cauliflower has been developed for. It's a resistant for. Options are given as option A, resistant to aphids, option B, low sugar content, and option C, resistant to blight, black spot, and option D, high nutrient crop. So now we see resistant to aphids. Aphids resistant, it is seen as a brassica, and low sugar content. Low sugar content is a morphological character of the plant to resistance of insects and pests, and resistance to blight, black spot. So Pusa snowball is a cauliflower variety, means disease resistance variety, that is resistance into the disease of blight spot disease, blight black spot disease, and curl blight disease. So answer is these are the Pusa snowball K1 is resistance into the blight black spot, otherwise curl blight disease. The resistance into blight black spot disease and the curl blight disease. And option 4, high nutrient quality. So these are the low sugar quantity and the high nutrient quantity. Those are the morphological, physiological, biochemical characters or changes are seen in plants to produce disease resistance varieties. And next question is, one of the organism commonly used as SCP. So SCP is a single cell protein, and these single cell proteins are the alternative source of nutrients for the human beings and animals. So some of the organisms are they can use as a food materials. So in these are the some organisms are they have identified. Option A, spirulina is a single cell organism. It's a cyanobacteria only. And option B, Agaricus red algae. So Agaricus is fungi we call as a mushroom, edible mushroom. It is Agaricus only, and it's not belonging into the red algae. It belongs into the fungi. So mismatch it's not correct. And option C they given as Saccharomyces. It's also called a scientific name of the yeast is Saccharomyces cerevisiae. Yeast it is. So they given as a bacteria. So it's not a bacteria, it's a fungi. And option D they given as an E. coli. It is Escherichia coli bacteria. It was present in a intestine it was present into the human beings, and these E. coli bacteria can maintain the insulin concentration into the human beings. So E. coli bacteria will causes the disease into the human beings also, and E. coli bacteria will not use as a SCP. So out of the given options, one of the organism only can use as a SCP, that is spirulina, which belongs into the spirulina. And next, second one, which amino acids are present in biofortified maize? So here the biofortification, one of the method we are seeing into the plant breeding, increase the nutrient concentration. By the process of plant breeding mechanisms, we can increase the nutrient concentration of crops, that is seeds or fruits, whatever it may be. So biofortification, the varieties are we call as biofortified varieties. So biofortified maize variety. So which amino acid? In these are the biofortified maize variety, which amino acids are will be present? So in the year of 2000, one of the new variety of maize was developed from the plant breeding mechanism. So in these are the maize amino acid concentration will be found twice than before. So those are the amino acids. Option A they given as methionine and tryptophan. Option B they given as methionine and lysine. Option C they given as a tryptophan and cysteine. And option C they given as a lysine and tryptophan. So now the answer is that is in the maize or biofortified maize variety, it contains as a twice number of amino acids than the previous. Those are the amino acids are lysine and tryptophan. Lysine and tryptophan, answer is B. And next one, 8. Which one of the following variety okra is resistant to shoot and fruit borer? So it is a insect resistant variety as a okra. Some of the okra it is resistant into the fruit borer and shoot borer. Means which variety of the okra it is resistant into the fruit and shoot borer. So they given as a Pusa Doro and Pusa Savani. Pusa Doro is a brassica variety, its resistance into the aphids, and Pusa Savani belongs into the flat bean. Pusa Savani and the Pusa A4 is common. Next question is 8th one. Which one of the following variety of okra is resistant to shoot and fruit borer? So okra is a insect pest resistant. For some of the insects, those are the insect pest resistant of okra variety we have to identify in the given options. Those are the given options of Pusa Doro and Pusa Savani. Pusa Doro it was belongs into brassica, its resistance into aphids. Pusa Savani it is belongs into the okra, and option B they given as a Pusa Savani and Pusa A4. These both are belongs into the okra variety only, and option C they given as a Pusa Sentu and Pusa Doro. So Pusa Doro it belongs into the brassica variety, and Pusa Sentu it belongs into the flat bean. It's not belongs into the okra. And Pusa Sentu also it belongs into the flat bean. Pusa A4 only is a variety of okra. So now the answer is which one of the following variety of okra resistant to shoot and fruit borer? For the shoot and fruit borer resistant variety of okra, it is Pusa Savani and Pusa A4. It's the answer B. Question is 9.1. The plantlets obtained from tissue culture are transferred to field under the controlled conditions or reduced light intensity and high humidity. The process referred as. So the question is, after the completion of tissue culture, means after the transferring of the callus explant into the nutrient media, from the explant undifferentiated mass of tissue was developed, that is called as a callus. From the callus, by the process of morphogenesis, will be get the plant body, means plant organs will become developed. After the morphogenesis, this plant we are going to transfer from the nutrient media, that is from the laboratory conditions, into the greenhouses. So at the time of the transferring of plants from the laboratory conditions or labs into the greenhouses to get the good light and by the changing of environment. So which that method it is called as changing of the plant body from the lab conditions into the normal environmental conditions, that is as a greenhouses, otherwise into the directly field. It's called as, options are given as subculture. So subculturing is a mechanism from at the time of tissue culture. We are going to separate the culture media from the tissue culture. From the culture media we will be going to producing as a new plant roots. So here the subculturing means we are going to separating down the culture media to produce a new plant root. But here the question was asking as, so what's the mechanism, what's the method we are going to use to transferring our plant body into the direct greenhouses, otherwise as a field. So subculturing, it's not a suitable method or it's not a suitable name for it. And next, B option, regeneration. So the regeneration mechanism, as early we are studying into the reproduction in organisms. So the regeneration means regaining of the last parts. And option C, hardening. See now, by the time of transferring of the plants from the lab conditions into the greenhouses, otherwise direct to the field directly. So some of the changes of the plant will be going to need it. The changes only we call as a reducing of the light intensity and the increasing of the high humidity mechanisms. At the time of the plants, they have to survive themselves. They are not adapted into the artificial environmental conditions. They have changed the artificial environmental conditions into the natural conditions. That is only we call as transferring of the plant from the lab conditions into the greenhouses, otherwise to directly to the field. That is we call as hardening. And option 4 they given as plant-led transfer. So after the formation of the plants, some of the plant are done, again they are going to use as a explant, will be going to use for the next production. That is called as a plant-led transfer. So here the question was asking, after the completion of the plant production, we have to transfer the plant body from the lab conditions into the greenhouses, otherwise the field directly. And that mechanism, that method is called as hardening. So answer is a C only. And next, 10th one, tomato is a somatic hybrid of. Now most of the questions will be going to get from the plant breeding, that is steps involved into the plant breeding, otherwise high yielding disease varieties or insect resistant varieties will be going to get the questions. And the other one is applications of tissue culture. So one of the application of the tissue culture it is somatic hybridization mechanism. So one of the somatic hybrid, it's a tomato. Tomato is a new variety. It was produced from the options are given as poppy and potato, potato and tomato, tomato and tamarind, and poppy and tomato. The correct answer is somatic hybrid of the tomato. It was developed from the two parental plants. The two parental plants are potato and the tomato. So that's why these two names are combined together. We will be going to call as tomato. The new variety of the tomato, it is developed from the two parental plants as a potato and the tomato. So these are somatic hybrid. So answer it is B only. And next one, which are these not necessary for tissue culture? Which are the following not necessary for the tissue culture in a nutrient media? Option A, they given as the nutrient medium should be sterilized. The nutrient medium should be sterilized. Tissue culture is a mechanism we are going to use into the production of new plant plates within a short period of the time. So now here, by the using of the that is in the tissue culture, nutrient media can play a key role. So in the option A, the nutrient medium should be sterilized. To performing of the tissue culture, what are the equipments are we are going to use? Those all are the equipments and those all of the nutrient media should be sterilized. Sterilized or sterilization means we are going to call as removal of microorganisms. So any microorganisms are present, otherwise any organisms are there present, they will be going to show the contamination that tissue culture will be going to will not give a new plantlets. properly. So we have to perform the tissue culture before only what are the glassware, otherwise nutrient media or other chemicals, otherwise water we are going to use. Those all are firstly sterilized. So here the nutrient media should be sterilized. So to the performing of tissue culture, nutrient media we have to sterilize. And option B, energy source usually in the medium is sucrose. So to the creation of nutrient media into the tissue culture, that energy source always will be going to get in the form of a sucrose only. And option C, the medium must be supplemented with the minerals, vitamins and amino acids. So we are going to create the artificial nutrient media for the tissue culture. That nutrient media we are created by adding of minerals and amino acids and vitamins. So what are the components are required into the plant? Those all of the components are we are going to added artificially instead of the soil, because we are not using as a soil in the tissue culture. We have to create the artificial nutrient media. So, and option D, medium must be provided with growth regulators like ABA. Here we are going to add it into the all growth regulators into the nutrient media. They are given as the growth regulators like as ABA. ABA means we call as abscisic acid. Abscisic acid is not a growth regulator, it's a growth inhibitor. So growth regulators are three. Those are RZ, gibberellin and cytokinin. So these three are called as plant growth activators or plant growth regulator. These three only they can use into the tissue culture widely to give the plant body from the explant. When you are going to add in the ABA, ABA is a plant growth inhibitor. It can inhibit the growth and development of the explant. So that's why ABA we are not used into the plant growth. Means ABA we are not used into the development of the plants. Means explant we have converted into the new plant plates. At the time of the plant growth activators only we will be going to use. This ABA can restrict the cell division, can prevent the growth and development. So that's why incorrect statement of the given options are D only. Remaining three A, B, C, those are we are going to use in the tissue culture, but option D only will not use. So that's why answer it's an incorrect statement. And next one, 12th, Sonalika and Kalyan are high yielding variety of. So we'll be going to produce the at the time of Green Revolution, semi-dwarf varieties of wheat and rice was imported from the different countries. So those are the semi-dwarf varieties of the wheat, Sonalika and Kalyan Sona. Sonalika and Kalyan Sona, those are the semi-dwarf varieties of wheat. They given as options as maize, rice and sugarcane. Out of the four options, correct answer is they belongs into the Sonalika and Kalyan Sona into wheat. These both are wheat varieties only. In the same way, rice varieties are there as the questions. Those are the high yielding semi-dwarf varieties of the rice. Those are Jaya and Ratna. So here they are asked Kalyan Sona and Sonalika. Those are belongs in the wheat variety only. Next question. Select the correct pair of difference between pure line selection and mass selections. Question from the steps involving a plant breeding to the performing of plant breeding. We have to select the proper parent, means select the parental plant. That is done by the two types, pure line selection and mass selections. Out of the given two types of the pure line selection and mass selection, which are the options correct I have choose. So you want to know the pure line selection and mass selections. In a steps involving plant breeding, we have to collect the or we have to select the parent. So those are the parental plants should be developed from the self-pollination only. So you know self-pollination means what are the parental plant characters are there present. Those are the characters are we are seeing into the offsprings also. So to performing the self-pollination or to select the pure lines, we have to perform the self-pollination as 6 to 8 generations. Until the completion of 6 to 8 generations, what are the characters are their presence in a parental plant. Those characters are after the completion of 8 generations also will be donated. Such type of the plants are we call as, or such type of the parental plants are we call as a pure line selections. And coming to the mass selections. So mass selections are also like as a pure line selections only, but here we have done or we are going to selected the parental plant body after doing the 6 to 8 generations. as self-pollination, otherwise the cross-pollination. Either the self-pollination or cross-pollination. Mostly in the mass selections we can select the parental plant through the cross-pollination. So if you are done the cross-pollination from the one generation to the next generation, many number of new characters will be going to introduce in a offsprings. So in a pure line selections, new characters will not develop or will not perform in the mass selections, in the parents, from the parents into the offsprings. So those are new characters will be going to introduce. So based on the pure selection and mass selections, option A is given as in a pure selections use both self and cross-pollinated species. Here we will use into the pure line selection as self-pollination, and mass selection used only self-pollinated species. So in the mass selection we will use into the cross-pollinated plant species. And option B, genetic variation is absent, means new characters are not transferred, are not introduced into the offspring. So that is called as genetic variations are absent. Here the genetic variations are present. Offsprings, what is the new variety we are going to produce from the parents, those offsprings will not get the new variations in a pure line selections, will get the new variation in a mass selection, because in the mass selections cross pollinations take place, but in a pure line selection self pollination only takes place. And option C, variety has wide adaptation. Variety has a wide adaptation. Variety has a narrow adaptation. Here the pure line selection and mass selections, here adaptation we are not considered as a criteria for the selecting of plants. So adaptation will be means modifications to the environment, it is to survive like that. So it is not considered the criteria for the pure selection and mass selection. And option D, production of variety is less uniform. Production of variety is high uniform. So here production of variety is less uniform, production of variety is a high uniform. In these are the pure line selection, production variety is high uniform. In the mass selection, production of the variety is less uniform. So that's the difference. So out of the four options, pure line selection and mass selections are pure line selections are we don't get the offspring of the specific plant through the self pollination only, variations are will not show. So that's why answer is. And 14th question, next one. A wheat variety Atlas 66 which has been used as donor for improved wheat is rich in. So Atlas 66, one of the biofortified wheat variety. So these are the wheat variety, it's contain as much concentration of biofortification is a mechanism to increase the nutrient concentration into the seeds or plants. So Atlas as one of the wheat variety, it's contain as much concentration of the components as option A iron, B carbohydrates, C proteins and D vitamins. So Atlas 66 is a wheat variety. It's contain as much concentration of or double concentration of proteins than the previous. So that's why answer is C. And question number 15. 250 grams of methylophilus and methylotrophus can be expected to produce dash types. So methylophilus and methylotrophus bacteria, one of the SCP. SCP single cell protein is an alternate source of the food materials for human beings and the animals. So we are taking as a 250 grams of methylophilus and methylotrophus bacteria. It can produce how much tons of food? So that is 250 grams of the methylophilus and methylotrophus bacteria can produce 25 tons of proteins. So it's a direct question only. So 25 tons of the proteins will be going to develop from the 250 grams of methylophilus and methylotrophus bacteria. In your books they given as one of the comparisons, you are taken as a 200 kgs weight of the trout can produce into the 25 grams of the proteins only, but we are taking as a SCP 250 grams of very less concentration of 250 grams of the bacteria can produce 25 tons of proteins. So that's the use of SCP. And next one, 16. What is not true about emasculation of flower while performing an artificial process? See now, emasculation is a mechanism, means removal of anthers from the bisexual female flower. So removal of anther from the bisexual female flower to produce a new variety of plants by the process of plant breeding, we are going to use as a two parents. One of the parent as a male and female. The female, it was there as a bisexual nature. The female can produce into the bisexual flowers. From the bisexual female flower, we have to remove the anthers. If anthers are not removed, they can perform the self-pollination. Here the plant breeding we are going to performing to produce a new variety by the using of cross-pollination. Emasculation was not done, means self-pollination is takes place. The emasculation was done into the bisexual female flower only. So option A, it is removal of anthers from a flower. Option B, it is done before anthers. Option C, it is avoid self-pollination. And option D, it is done in flowers of plants selected male parent. So see now, already I said it is emasculation was done into the bisexual female flower. But here option D they given as it is done in a flower of plant selected male flower. So if the flower is male, it's contained as a only anthers. We are no need to done into the emasculation mechanism. Because we are performing to the cross pollination. At the time of the cross pollination, bisexual female flowers are there, means in the bisexual female flower to avoid the self pollination we have done the emasculation. So incorrect statement of the given follows is D only. Answer is D.